Hello, everyone. Welcome to another installment of Paleo Perks. We're just going to wait a few more seconds to make sure everyone has been able to join the meeting. We have a few more people trickling in. Okay. Okay, and I'll go ahead and get started. So hello, everyone. Welcome to the 26th of November, 2024 installment of Paleo Perks. Today, we will be hearing from Peter Hällberg from the Department of Geological Sciences at Stockholm University in Stockholm, Sweden. And so just a little bit about the format of today's seminar. We'll start out with welcome and announcements, and then there will be the main talk for about 30 minutes. After the talk, there will be a moderated question and answer session for about 10 minutes. If at any time during the talk or during the question and answer session you have questions, please send those questions via the chat function to the questions@PaleoPerks host. So first, a little bit of Paleo Perks housekeeping. Paleo Perks values the participation of all folks interested in the paleo sciences. Please remember to abide by our code of conduct during today's seminar. Please mute yourself for the duration of this talk. You should, by default, be muted, but if for some reason you are not muted, please mute yourself and try to stay muted in order to respect our speaker today. Again, if you have any questions, you can chat with the questions@PaleoPerks host, or alternatively, you can use the raise hand function. If you have any technical issues, those should also go to the questions@PaleoPerks host. We have closed captions built into Zoom. You can use the CC button to show or hide them. If you have an early career researcher who you'd like to nominate to give a Paleo Perks talk, you can do so at this link here. And we would appreciate it if you would fill out our weekly feedback form for demographic information. It's totally anonymous, it's totally optional, but it is encouraged, and it really helps us shape the seminar in the ways that can best serve the paleo community. And you can find that link in the chat window as well. So a little bit more about today's speaker, Peter Hällberg. He did his bachelor's in earth science at Uppsala University in Sweden, went on to do a Master of Science in Physical Geography, also at Uppsala University, then went on to do a PhD in Geology at Stockholm University in Stockholm, Sweden, and is currently a postdoc at Stockholm University in Stockholm, Sweden. So with that, I would like to hand it over to Peter and enjoy the talk.
Thanks a lot. And I hope everyone can hear me now and see my screen. Good. So first of all, thanks a lot for having me here and for letting me speak. I'm really happy for that. And thanks everyone who's come in here as well to listen. And yeah, I hope we can have a good discussion also at the end. So I'm Peter Hällberg. And I finished my PhD earlier this year at Stockholm University. And what I will present here is a little bit of the findings from the PhD. And what I will talk about is seasonality and how we can reconstruct seasonality in the tropics. And this talk is about disentangling then the seasonal and annual precipitation signals in the tropics over the Holocene using delta D, alkanes, and GDGTs. So when we see these two pictures, we can immediately see that there is a big difference in the climate here. And with the savanna being much drier than the rainforest on Sumatra. But I will actually argue in this talk and try to convince you that it's not the total amount of precipitation that is the most important here, but actually the seasonal distribution of precipitation. And if we look at these climate diagrams for these two locations, we see that the annual precipitation is very high in both locations. But on the savanna, we have a very, very, very dry winter period. And that is the main thing that's driving this difference. And when I talk about seasonality, I'm talking about the amount, timing, and duration of precipitation. So basically, if rainfall is spread out evenly over the year, or if it occurs during a specific season. And as we saw in the previous picture, this can be very important for ecosystems, but also for agriculture and human societies, etc. And the location that I've been working on seasonality is on Sumatra, which is located in the Indo-Pacific warm pool. And this region has been called the heat and steam engine of the world because it exports very large amounts of moisture and heat to the extratropics, and is one of the important drivers of the monsoons. And temperature seasonality in the tropics is fairly low, and at this site it's around 1 degree only over the whole year. But precipitation seasonality is much bigger. And this is driven by wet and dry monsoon seasons. And if we look at these two figures, so these are the wind patterns. We see that in northern hemisphere winter, we have winds coming from the north and northeast. That's related to the Asian winter monsoon. But in the northern hemisphere summer, these patterns are completely reversed. So the seasons are very pronounced in terms of atmospheric circulation. And this circulation in this region is driven very strongly by the intertropical convergence zone. So the intertropical convergence zone is a zone of maximum convection and precipitation. And this migrates with the season towards the warmer hemisphere. So in the northern hemisphere summer, the ITCC is shifted towards over mainland Asia. And in the southern hemisphere summer, it shifts south. And this also is what drives these near surface winds. And over or in the ITCC, we have very warm, humid air that's rising, and this is what drives a lot of precipitation and monsoons. And if we look at precipitation in Sumatra, in this bar chart, we see that precipitation is strongest in autumn and spring. And this is during the passing of the ITCC over the region. So we have these two peaks in precipitation. And this region is also very interesting and important because the heat and convection in this area drives this east-west circulation that's called the Pacific Walker circulation. And this is one of the important drivers of El Niño Southern Oscillation. So what happens in this region really is very important for global climate variability. And when we go to paleo stuff and paleohydrology reconstructions in this region, there already are several, mostly based on speleothems, but also from leaf wax isotopic reconstructions. And what has been found is that for the deglacial and the last glacial maximum, we had much drier conditions compared to present day, with the driest and coldest period around Heinrich Stadel 1, around 16,000 years ago, and then warming and more wetter conditions over the Holocene, peaking somewhere in the mid-Holocene. And this reconstruction and other reconstructions as well, they reconstruct the amount of precipitation. But my question in this talk is then, what signal does the proxy actually capture? Is it the annually integrated signal, or is it a monsoon signal when you have the most precipitation that's recorded, or is it some type of dry season signal? As well as what is the impact of changing seasonality? And here I just overlaid over the same time period climate model simulation data for the same time period from the model trace. And this shows that there was a huge change in seasonality over this time period, especially in the late glacial and early Holocene, both in terms of temperature and precipitation. But seasonality is very challenging to reconstruct with proxies. And what has been used mostly in the past is different types of vegetation proxies, where a shift toward more grassland versus forest vegetation is interpreted as a more seasonal climate. And when there's forest vegetation, it's more stable and probably wet conditions. But it's hard to disentangle if it's only wet and dry or more seasonal and less seasonal as well. But this has been done using pollen and 13C mostly. And another thing people have used to infer seasonality is wildfires. And I think both these different methods are very good and can really give information about seasonality. But there are several drivers of both wildfires and vegetation, and it's hard to constrain what is actually seasonality and what can be other drivers. So in my opinion, a big knowledge gap here is that seasonality has a huge impact, but we are not able to reconstruct it well. And this is because we are lacking methods, and it's quite challenging. So what I'm trying to do in this study is to reconstruct Holocene precipitation, and also trying to tease apart what is seasonal signals and what is annual signals in proxy records. And to do this, I use a peat core from Sumatra that covered the last 8,000 years. And using lipid biomarkers, and then also comparing these to our results to climate model simulations. So in terms of methods, I've been reconstructing N-alkane distributions and isotopes. So N-alkanes, they are straight chain hydrocarbons, and they are very abundant in the environment. And they are produced by as leaf waxes by plants, and they're also produced by algae and bacteria. And the chain length distribution of alkanes are specific for different biomass types. So algae and bacteria mostly produce very short alkanes, so C19 and shorter. Aquatic plants produce longer N-alkanes, up to about C25, maybe also C27. And terrestrial plants produce the longest N-alkanes, where there can be some distinction that grasses produce C33 and 35 more than forest vegetation. Yeah, so what we reconstruct is, or what we measure is the distribution, but then also the hydrogen isotopic composition of these N-alkanes. And hydrogen isotopes in precipitation, so we have two stable isotopes, deuterium and protium. And deuterium is heavier than the most common hydrogen isotope. And this heavier isotope is less prone to evaporate and more prone to condense and rain out. So what happens in nature? So the ocean water is defined as delta D 0, and water vapor, not as much deuterium is taken up in water vapor. So that is depleted, and then the longer distance this air parcel or moisture travels, and the more rain out there is, the more negative the isotopic composition becomes. And in the tropics, the most important effect here is the amount effect. So the more precipitation there is, the more depleted the isotopic composition of water will be. And then there are some complicating factors like changes in moisture source and trajectory, and the amount of evaporation can affect this. But mostly the amount effect dominates in the tropics. And then to get from delta D of water to delta D of leaf waxes. So water falls on land, and then vegetation takes up this water. And during biosynthesis of lipids, there is a fractionation. So the values we get are much lower than what's in the precipitation. But these lipids then, the hydrogen isotopic composition of these lipids reflect the delta D of the precipitation during growth of these plants. So what we are using in this project was to look at the delta D from different chain lengths of N-alkanes. And the delta D from alkanes produced in different seasons then allow reconstruction of precipitation seasonality if these alkanes are produced during different seasons. And we also used GDGTs, so glycerol dialkyl glycerol tetraethers. And these are microbial membrane lipids, both produced by bacteria and archaea. And the relative abundances of these different GDGTs have mostly been used to reconstruct temperature. But they are also very sensitive to other environmental factors like pH, moisture, source environment, oxygenation, etc. But they are very good to use or convenient to use because the GDTs are abundant in most environments globally, whether you're looking at lakes, oceans, soils, or peats, etc. And these peat cores that we used, so these peats, that's partially decomposed organic matter, and they occur where the organic matter production is faster than the decomposition rate. And you basically need slow decomposition, and that often happens when you have waterlogged conditions. But we have very slow and continuous deposition of these peats, about 1 millimeter per year, allowing us to reconstruct conditions back in time. So for the results, first of all, alkane distributions. So these are the average alkane distributions through the whole core. And what we can see, so we have a lot of C29 and C31. So we have a lot of terrestrial plants producing alkanes. And we also have a lot of aquatic plants, so these mid-chain alkanes, as well as a lot of C19, which most likely is produced by algae. And if we look down core over time at these distributions, so this is the amount of aquatic plants. And aquatic plants grow throughout the year or throughout most of the year. So we interpret this to reflect the annual wetness of the peat, and this peaks around 3,500 years ago. That's when we had the wettest conditions, according to this proxy. And algae, so the C19 alkane, they have shorter life cycles and grow during flooding of the peat, so when it's very wet conditions, and probably also when there's nutrient input. So we interpret this to reflect floods. And we can already here see, so aquatic plants and algae, they both require wet conditions, but the timing of their peak is different. And I will get back to that later. And then in terms of isotopes from the N-alkanes, so from terrestrial plants, so C29 and 31, So this is the proxy that is most often used. These when leaf wax isotopes are presented, either one of these are usually the one that's used. And that's because these plants grow throughout most of the year, so these likely reflect an annually integrated rainfall amount. Whereas the delta D from the algae, since they grow during the wet season, they most likely reflect the wet season rainfall. So we interpret this as a monsoon signal, and we can see here that it's fairly different when they indicate wettest conditions. So the monsoon signal here, all the isotopes from the algae, indicate wettest conditions from 4,000 to 6,000 years ago, whereas the terrestrial plants indicate increasingly wet conditions all the way until about 3,500 to 4,000 years ago. So if we take the difference between these two, so basically the difference between seasonal and annual rainfall, we get a precipitation seasonality index. So this delta D terrestrial minus algae. And then we see that the earlier parts of the mid-Holocene was more seasonal than the later part of the Holocene. In terms of GDGTs, so first of all, this PGDT is a precipitation or humidity proxy that was published earlier this year. And that indicates that we have the wettest conditions between 3,000 and 4,500 years ago. And then crenarchial, so that's a GDT that's produced by ammonia oxidizers. So for the production of crenarchial, you need some oxygenated conditions, so drying out of the peat. So we interpret this as droughts in the peats. And then a synthesis to put all these results together. So first of all, in terms of seasonality, we have an indication of droughts between 4,000 and 6,000 years ago from crenarchial. And at the same time, we also have indication of floodings and a very high algae production. And in terms of delta D, so the precipitation seasonality index, we also see a higher seasonality in the earlier part of the record. In terms of fires, so we also have a fire reconstruction from this site, and that's done via levoglucosan, which is a biomass burning product. We also have increased wild biomass burning in this period. So what we have between 4,000 and 6,000 years ago is very strong seasonality. So we have both droughts and flooding at the same time, as well as fires, probably during the dry season, as well as increased precipitation seasonality. In terms of annual conditions or annual wetness, the peak in when we have the wettest conditions occur slightly later, about 3,000 to 4,500 years ago. It's indicated both by the delta D of terrestrial plants, the abundance of aquatic plants, as well as GDGTs. So yeah, peak humidity between about 3,000 to 4,500 years ago. So then the question is, why do we have this strong seasonality in the mid-Holocene? And one option or hypothesis would be that it's insolation, because insolation is an important driver of precipitation in this region. And so in this figure, we have age on the x-axis, and on the y-axis, we have different months, and the color is insolation. So dark colors is strong insolation, and light colors are low insolation. So if we compare the modern climate, so in this column, we have low insolation during June, July, and then stronger insolation in both the autumn and spring. And that's more of the wet season, and this is the dry season. And if we compare that to the mid-Holocene, we see that this is quite different. So then in autumn, we had even stronger insolation, likely than driving precipitation, stronger precipitation in the autumn months, and weaker insolation in spring. So the hypothesis that we had here was that, okay, we have a more intense precipitation in the wet season, so autumn wet season, and then this dry period in the summer and spring is made much longer. So then we tested this using a climate model. So this is the seasonality proxy, delta delta D. And if we overlay the autumn minus spring insolation here, it follows this very closely. And also, so we tested we tested this or compared this to climate model simulation data with MPI ESM 1.2. So this is a transient model that runs over the last 8,000 years. And that shows very similar results to what we see in the proxy, and also similar to insolation. And what it shows, so here are also as maps. So autumn precipitation compared to so autumn precipitation in the mid-Holocene compared to the last 2,000 years, and then spring precipitation. And what we see is that the wet season is indeed intensified, so much wetter conditions in the autumn in the mid-Holocene, and drier spring periods. So yeah, so this points to that insolation was the most important driver of these changes in seasonality over time. And I also want to make a comparison to a speleothem record, because speleothems are kind of the golden standard, in my opinion at least, for past hydroclimate reconstructions. So our record shows the wettest conditions at around 4,000 years ago. And the speleothem, and this speleothem is from only like 100 kilometers away from our site. That looks fairly similar with a wet mid-Holocene and then drier late Holocene, as well as drier in the early Holocene. But the timing for the peak precipitation is quite different. And if we then instead show our monsoon reconstruction, we see that this fits almost perfectly together with the speleothem. And this offset between delta D and so delta D from leaf waxes and speleothem 18O is also seen elsewhere in Indonesia. So an open question that arises from this is if these equatorial speleothem records rather reflect monsoon strength than annual conditions, as they are usually interpreted as. Yeah, to summarize, we had the overall wettest condition between about 3,000 to 4,500 years ago. And we have evidence for intense seasonality earlier than that, around 4,000 to 6,000 years ago. And yeah, I think very intense seasonality in the mid-Holocene is not something that has been reported before from this region, at least. Yeah. So there are, of course, uncertainties with this kind of reconstructions, and I think the main one, so the main assumption that we have that is poorly constrained is that the alkenes from algae and higher plants are produced in different periods. And the growth periods and seasonality in this biomarker production is not well constrained. So one thing that could be done to make this case stronger and to maybe be able to develop a proxy that could be applicable to many different locations and not just this, would be to actually work with more modern samples to constrain the growth periods of different biomarkers. So to sample both in the wet season and the dry season, because likely we have quite different biomarkers produced when it's very wet compared to when it's very dry. So what biomarkers are produced in certain seasons and what are their abundances and isotopes? Yeah, so I've been writing a proposal on this and trying to make this more robust and also try to develop new methods to reconstruct seasonality. So if anyone has ideas how this could be done or things to look at, I'm very open to, or I'm very interested to hearing your suggestions. So take-home message from this talk is that multi-proxy biomarkers and their isotopes can reconstruct seasonality. And I think that this approach adds much nuance to previous isotopic reconstructions that have been looking at the total amount of precipitation. But here we can add more information to this. And with that, I would like to say thank you all for listening.
Awesome. Thank you so much for that talk. That was super interesting. Just a reminder to the people in the audience that if you have any questions, you can send those to the questions at PaleoPerks host. But we already have a couple questions. So Peter, if it's all right with you, we can just hop right into the question and answer.
Yeah, please.
So the first question, I'll post it in the chat, but I'll also read it out loud. How deep in time could you apply these methods? Do you have any interest in looking at deeper time?
That's a good question. So alkanes have great preservation potential. So I mean, I don't think there is really a limit to how far you could go back. Millions of years for sure. Probably many millions of years. So like you could look at deep time as well when it comes to these alkanes. GDGTs, I think, should also be possible to go really deep in time. Levoglucosan will probably degrade too much because I'm not sure actually because I haven't looked at these very long time scales, but I think that's as a molecule is much less stable than especially alkanes and GDGTs. But yeah, I mean, it would be very cool to apply these methods to all different types of time scales, and with alkanes, I think it would be very possible.
Great, thank you so much. We have another question that has been submitted. To what extent did vegetation changes, C3 versus C4 photosynthetic pathways, contribute to the delta D of N-alkanes during the mid-Holocene compared to the influence of annual precipitation?
So we have the 13C of the bulk, and that does not change almost anything, or it changes very little when we see this change in seasonality. So I think it did not have a big impact on this. So it would have been very nice to also measure the 13C of the alkanes themselves, but we had some problem with the instrument, so we didn't do that. We used up everything for the delta D. So unfortunately, we cannot constrain it very well for the alkanes, but we have done it on bulk, and then there we don't see anything. Yeah.
Thank you. Okay, another question. Okay. How many seasons can you distinguish in the reconstructions? Is it limited to 2 seasons, wet and dry, or could you potentially see 4 seasons or more elsewhere in the world?
So this really depends on when your biomarkers are produced. I think in our case, we cannot constrain it well. Then we would actually have to go out there and, as I said in my last slides, to constrain this more and see what is growing in different parts of the year. In theory, if you investigate this and you have different biomarkers produced by different types of organisms over the year, then it would be possible to distinguish. But someone has to do this work as well to actually investigate this, and it hasn't been done yet. But I think it would be really cool if the community would do that.
Yeah, definitely. That would be very interesting. Okay, we have lots of questions coming in, so
Nice.
I think it was definitely a very interesting talk. So this next question, insulation is important, but is it local insulation or that over remote regions? Which latitude is the insulation in your plot at? Thanks.
So the plot is from 2 degrees south, so the same as our location. So yeah, so this site is just south of the equator, and our site and the plot is as well. Yeah, I think it doesn't make a huge difference which region you take, because the insulation, the absolute number changes, but the trend over the Holocene remains the same if you're on the northern hemisphere. Yeah.
Thank you.
So I think this, like what we see in our record, it ties together to what happens like in the high latitudes as well in the northern hemisphere. So in the mid Holocene, you have these very warm summers compared to the late Holocene, and I think that is one of the drivers in this as well, because that will affect the monsoons. Yeah.
Thank you. Okay, next question. Were there human activities around the region during the mid-Holocene? Could that have impact on alkanes or fire records, etc.?
Yeah, definitely. It would be difficult to say. I think you could see it in pollen reconstruction, like if we had pollen from this site, which we don't. But yeah, I mean, if people go there, start to drain the peat or alter the hydrology, then for sure that would affect what we see. And I guess that's a problem you often have in paleo research in the Holocene. Like, people are starting to pop up and they are altering the environment. But we haven't seen any evidence from that here. And like, since the 13C, for example, doesn't change a lot, I think if you would change the environment a lot, then we would probably have seen that. And what we see in terms of 13C, for example, is a huge change in the last few hundred years. So I think what we see in our core is that in the last few hundred years we have a big impact from humans, but we don't see anything earlier than that. But of course, there could have been people around and altered this somehow. But yeah, it's hard to constrain, I think.
Definitely. But that is a really interesting
Yeah, it is. Yeah.
question.
Yeah. Maybe I can say we have also another peat core from Sumatra, and there we see a big change around 8,000 years ago, and we were a little bit suspicious if that was related to humans, and we also saw a lot of more rice pollen, so some early rice, some early form of rice, and that could have been like early farming, and that my colleague is working on that. But that could have been humans starting to affect the area. But yeah, I mean that is ongoing work, and if it would be rice, then that would push back the arrival of humans and or not the arrival of humans, but the start of agriculture by several thousand years. So I mean, I'm not going to say that that's what happened, but it is possible.
Awesome, thank you. Okay, our next question. This person says, pretty curious open question in the end. What are your thoughts about the time lag between speleothem and leaf waxes?
So I think speleothems, like it's very hard to constrain when in the year their signal is captured, and so it's hard for speleothem researchers maybe to answer this definitively, but I think like from what we see that the speleothem record is very similar to the algal delta D record, shows that it is more of a monsoon signal in the speleothem, and leaf waxes, so the terrestrial plant leaf waxes, they could then grow over more of the year, so capture more of an annually integrated signal, even though there's probably some type of seasonal bias also in the leaf waxes. But yeah, I think it's a difference in when they take up the water, basically.
Great, thank you. It looks like we still have a couple questions left, Petter, if you have time for a couple more.
Yes, please, yeah.
Well, this next one is an easy one because it's not actually a question. So
Okay.
It's just a comment, but I thought I'd just share it. This person, and actually this was submitted from Elise H. They say, not a question, just a comment. Thank you for explaining how you are using DH and alkane biomarkers in a way that was easy to follow. And I think I agree. I think you did do a good job of explaining it. So I just wanted to put that out there.
Yeah, thanks a lot. Thanks.
Okay. Here is a question. Okay. This person says, thanks for the very interesting talk. I have a question about the regional setting of this peat site. Is it like a pond in the wet season? Because I am wondering about the water source related to delta D of the algae. Do they use the water from the pond during the wet season instead of rainfall?
Yeah, it could have been nice to have a figure maybe of the site. So this is a peatland, so it's a valley swamp, and there is a pond close to the site, and this pond is very variable in water depth. So as I've been looking at old satellite images, and sometimes it's completely dry, and sometimes it's a lot of water in it. So the whole site would be flooded. So this is a very interesting question. So I think this site is not like one of these high latitude peats where you only have water directly from precipitation, but this precipitation will also be more kind of this pond, as you say in the question, rather than directly the rainfall. So it will be a rainfall that has been sitting around there for a while, and that could of course also cause some more evaporation, and that would see an enrichment of the delta D. But so what we see here, I mean, this is not due to evaporation or things that are happening in the pond. That would have had the opposite sign. So I don't think you can explain what we see in our record in terms of isotopes from this. I think that would have the opposite effect. But it's a very interesting question, and if you have any more comments about them, please let me know.
Great, thank you. It looks like we have 2 more questions. So our second to last question. Does the ENSO have any sort of control on seasonality during the mid-Holocene rather than the seasonal insolation changes?
This is also very interesting because ENSO is supposed to be very inactive during this period. ENSO, so we have huge changes in the hydrology in the late Holocene. So around this period, around 3,000 years ago, our record and many other records show very intense drying. And around 2,000 years ago, let's say 1 to 3,000 years ago, the ENSO is really starting to become stronger. So you have much more variability. So both very wet conditions, probably in some years, and very dry conditions in other years. And then the question, why don't we see ENSO in our seasonality reconstruction? I don't know. Apparently, it's like our proxies are not sensitive to this type of like weather events, because ENSO changes on like a 2 to 7 year time scale, and I think what we see are the annual changes. And I mean, yeah, it would make sense to think that we would have more like ENSO variability in this time period where we see more seasonality, but I don't think that's the case. Like from many other proxies, this is the time with least ENSO activity in a very long time, like since the LGM basically, at least. Yeah.
Thank you. Okay, the next question. Also, we did get a couple more last minute questions. So Peter, I know you might be kind of crunched on time. So if you have to go, that's totally fine. But
I can stay for like 15 more minutes. That's fine.
Okay, great.
Yeah.
Then I think there's definitely some interest in the audience because
Great.
we're still getting questions. Okay, but this question here. Does wildfire influence the delta D of N-alkanes through the thermal degradation of organic matter? And that was submitted by Tamizara-san S.
I have seen a paper that says that biomarkers are very strongly influenced by biomass burning, including the isotopes, that you have actually fractionation during burning. But that paper, if I recall correctly, was based on samples that they actually put in an oven to combust. And this levoglucosan that we see here is I mean, the fire that we would have had is much smaller, or the fires. So I think it's not really comparable. And I don't think we would have a big impact on our proxies. But it is possible. So if you have a fire like right on top of your site, it will affect the biomarkers. And if you are interested, you can send me an email and I can try to find that paper and send to you. Yeah.
Great, thank you so much. Okay, our next question. Okay, the next question. Did you average your D2H of C29 and C31 for the delta D terrestrial? Do you have plots of the individual alkane? And are there differences between the two throughout the entire records? Thanks.
Yeah. Yes, for this presentation and in the publication we did that, but we also plotted all individual alkanes in the supplementary of this paper in QSR. So if you're interested, you can check that out. But it was very small differences in all these long chain alkanes. Maybe I can. Yeah, if we go back to this one. So the best ones we had were 29 and 31, like they were in the highest concentration throughout the record. But if I recall correctly, 27 all the way up to 31 were very similar. And then, yeah, this is C19, and we also had very good data for that. But then for these mid chains, they were kind of in between how the algal and the terrestrial plants looked. So it looked like, so the algae, they had the most seasonal signal, and then the aquatic plants somewhere in between, and then the terrestrial were the most, yeah, they had the latest peak. But yeah, so I don't think it would matter if we showed C29 and C31 individually or averaged. Yeah.
Great, thank you so much. And now for our last question. Short and sweet. Since someone mentioned ENSO, how about IOD, Indian Ocean Dipole?
Yeah, when you're talking about ENSO, that's definitely something to think about as well, because they are correlated as well. I mean, I have not looked into the IOD so much on this, but I think the activity is similar to ENSO. But the person who asked it maybe can send me more questions later, because I don't have a good answer for this. Or if you think something is interesting about it that I should check out, then please send me an email.
Well, great. Thank you to everyone in the audience for your questions. And Petter, thank you so much for your awesome talk. That was super interesting, and I think I'm not just speaking for myself when I say that, because clearly the audience was very engaged and had a lot of good questions. So thanks again. Thanks to everyone for joining us today. Just a reminder to please fill out the weekly feedback form linked in the chat so that we can learn a bit more about who attended today's seminar. Again, it's totally anonymous, but that just helps us at PaleoPerks make these seminars even better for the paleo community. And just a reminder that next week you can join us to see Neda Shalev from Hebrew University in Israel present on tracing past oceanic magnesium budget with magnesium isotopes. So as always, you can find PaleoPerks on social media, and you can get in touch with us on our website. And yeah, we look forward to seeing you again next week. So thank you, everyone. Bye-bye.